First of all, thanks to organizer for giving me this opportunity to talk here. Today I will talk about protein search process, how protein search for the target site and do the functional job inside nucleus. If I think about this process, a very simple picture will be there are proteins, it wants to find one target, and very simplistic picture that it binds to the target and gene expresses and gives rise to this mRNA expression. This is the most simplistic picture we can think about. Now, there are models how this process happens, and people thought about this process that in the genome, a protein can move on the DNA and it can also do the 3D diffusion, and together that can give rise to a minimal search time and it can find the target very quickly. So this in between region where protein is doing 1D and 3D, there the target search is very fast. But this process is called facilitated diffusion, and mostly this has been proposed for the prokaryotic system. Now, if we think about the eukaryotic system, this proposal may not hold for multiple reasons. First, I will come to the experimental observations, recent experimental observation on this. So, as you see, this paper proposes and many other articles that protein spends most of its time in contact with the DNA. That means there are not much provision for the 3D diffusion in the cell, in the nucleus, and also the eukaryotic nucleus is highly dense and viscous. So protein will not have much scope for the 3D excursion inside nucleus. So one thing we are concluding from this experimental observation that there is not much scope for the 3D diffusion. Then what is the organization, chromatin organization, how it is helping the search process? That is one of the central question of today's talk. So we will concentrate on one of the functional subunit of the chromatin, which is topologically associating domain, and how that structure is helping the search process. Am I audible? Okay. So now, if you think about one single TAD, there are multiple loops inside TAD, and that is helping in the search. Whether that is helping in the search process, that is our question here. And these TAD boundaries are reached with this H3K4 MET marks, which marks the promoter region. And if you see this chipsec data of polymerase 2, POL2, you will see that there are enrichment of POL2 near the boundary sites. So these boundary regions are the target sites for the proteins, and how this internal structure of the TADs is helping in the search process, that is we are going to study. First we will do a simplistic model, and then we will go to more realistic model. So first we will consider a network-like structure where the contacts between different regions of the TADs can happen with a probability of P, and the nearest neighbor of those networks are always connected to maintain the chain connectivity. Now, what is that P probability of contact between different subunits inside TAD? So that follows a power law because many experimental evidence that the contact probability shows a power law with the genomic distance. But these powers, this comes for whole genome level. But in the TAD level, what is that power? So that power defines the compaction of the region. So if you start from here, there you can see gamma is equal to 2.2. And as you go more compact domain, your gamma will decrease. And these are some values of gamma that self-avoiding work has gamma of 2.2, random work 1.5, chromatin as a whole genome organization 1.08, but TADs gamma is less than 1. So we write an equation, master equation for the search process, starting from the ith site, what is that mean time to reach boundary sites. And first we consider there is no unbinding from the DNA, purely 1D motion on the DNA. And we ask the question, what is the average time to reach the boundary sites or the target sites? And we see a non-monotonic behavior as a function of gamma. That means when your region is very open, this side gamma is equal to 2.2, your time is large. When your gamma is low, like 0.4, 0.5, there also your time is large. Somewhere in between, you are getting an optimal time. So depending on the gamma, depending on the compaction, you can modulate your search time and thus the expression of gene, protein function, etc. Next, we move to when your P off is not equal to 0, you have a 3D diffusion. And in that case also, if you see that 3D diffusion, so this color phase plot is y-axis is P off, unbinding probability, x-axis is gamma, compaction. So low gamma compaction, P off is also low, then only you are getting a low search time. So in the nuclear environment where your 3D organization is very messy, there your unbinding probability low is giving you actually a low search time. Then we move to real TAD analysis. So we extracted all TADs from the human genome data, and we characterized all TAD based on their compaction and the length. So for each of these TAD, we characterize what is the gamma. So we take out the data for each TAD, we fitted with a power law and extracted what is C and gamma for each TAD. And those gamma has an average of 0.7, which is less than 1 as expected. And for each TAD, we have a length and gamma. So our characterization of the TAD based on these 2 points, gamma defines the compaction and length is the how long is the topologically associating domain. And from our earlier study, we have this phase plot. Y-axis is length, x-axis is the compaction, and this bluish region denotes the lower search time. Now we want to see that our actual TADs, where are they? And if we paste all the gamma and L from collected from the human data, they comes near the minimum of the search time. So human genome is organized in such a way, these functional subunits, small subunits, TAD-like regions, they are organized in such a way that your search time is already minimum. Moving forward from this point, how fast is the search? So if you have only a diffusive search process, you will expect that your time will go as L squared, where L is the length of your region. So we started with constructing a network from the high-C data. We put this contact according to the high-C data, Pij, and simulated the search process on this network, and we got the time. And in another approach, we characterize all the gamma, wrote this equation and solved this equation to get the search time. And in this case, the reds one from this approach from the simulation, and black is from this analytical calculation, and they matches pretty well. But the slope shows almost a super diffusive behavior. That means, and the L square behavior is here. This you will expect if your protein is only diffusive. Your protein is doing only thermal diffusion, then you will expect this scaling. But here, chromatin structure is giving the protein an advantage. If protein is following the chromatin structure, then it is almost moving like a super diffusive particle, and your time is minimum. So these are the two results. I will summarize now. So target search can be regulated by the polymer structure. All the earlier studies mainly focused on that how protein is moving, but they didn't focus on much on the polymer structure. Here we are taking a polymer structure from the high-C data, and consistent to that we are showing that the polymer structure is actually helping the search process. For the eukaryotic genome where density is high, there we are showing that 3D diffusion may not be very helpful. Contrary to that, there can be intersegmental hops which can help in the search process. And this TAD, all the TADs, they are not very compact, not very open. They are somewhere in between where the search time is minimum. So that's how genome is giving opportunity to minimize the search time. This is you can find this work in the bioarchive, and this work is done with Mithun and Ranjith and the project student others, and these are the funding. I'm happy to take questions.
Thank you, Shuvadip. Questions?
Yes, what could be the experiments that you can do in order to validate your model? So let's say if you rip out cohesin, for example, in some cells, what would be the prediction of your model regarding the regulation or for transportation.
So if you knock out cohesins,
Cohesin, so basically you remove the TADs.
well, if you knock out cohesin, your TAD structures are gone mostly. So your TADs will fuse, and these boundary regions which are getting more exposed due to the TAD structure, they are getting more contacts. That will not happen. But other than that, what I can think about that in vitro experiment, that you have DNA strands and you are putting some protein which compacts DNA, and you vary the concentration of that protein and measure what is the expression time. That can be one experiment, that whether with the concentration of that protein, whether your expression time is varying or not.
So thank you very much for your interesting talk. And I'm wondering, the collapsed globule has much less DNA accessibility to the proteins.
Right.
So did you consider this kind of accessibility
Well,
for searching process?
yes. So you are right that if it is a compact globule, there won't be much accessibility for the protein.
Yeah, penetration rate must be very low. Right?
Yes, but here, because our beads are coarse-grained beads, really compaction doesn't mean. So protein can really go through the coarse-grained beads.
Okay, I see.
But it's true that if your region is very compact, you will have multiple contact, and your protein will hover around many points. So in any point, you will have multiple contacts. So it will jump here and there, and that will make the process very difficult.
Okay, I see. Thank you.
If not, then let's close the session and thank all the speakers for giving interesting talks.